to the 38th Public Colloquium of Kadir Has University. This time we have a guest connecting to us from all the way from Massachusetts, and welcome Professor Daniel Aalberts from Williams College, Department of Physics. The title of his presentation today is Somewhere Over the RNAbow, Optimizing Protein Yield, mRNA Vaccine Technology, with a song in his heart. So I'm sure he'll talk more about the song part. He'll inform us concerning that bit. But first, I'd like to pass the microphone to our professor, Nihat Berker. Buyurun hocam.
Hello, everybody. Welcome. Welcome, Daniel. A warm, hearty welcome. It's a special pleasure to have Professor Daniel Aalberts as our colloquium speaker. Daniel has been a long-time friend and collaborator of mine, very long time. He hasn't changed a bit. I have maybe changed, but he has been a physics collaborator, and he's a great physicist, but also a lead artist in the performing arts, a person who's always upbeat, always smiling, someone from the very beginning who I have taken as a role model for performance, for being positive, for being happy, and for being a very close friend. Daniel, thank you very much for being here. And now I also would like to thank very much, as I always do, our president, Professor Dr. Sondan Durukanoğlu Faiz, for making these colloquia possible, a wide range of highly attended colloquia from which we benefit a lot. So thank you, Madam President, and I pass the word to you.
Thank you, Nihat Hocam. As always, I thank first Nihat Hocam for making this colloquium series as the brand of Kadir Has University. And of course, it's our great pleasure to have Daniel Aalberts as today's speaker. And I would like to give a brief information about Daniel's academic life, and then I will leave the floor to Daniel. Daniel, he got his BS degree from MIT in 1989, and then PhD, and guess what? With whom he did the PhD? With Nihat Berker at MIT in Physics department. Of course, he got the PhD in year 1994, and after that, he had several postdoctoral studies at University of Leiden and Rockefeller University. After that, he became a faculty member at Williams College and then chair in physics department at Williams College. And he has several honors. And now he is a full professor at Williams College. And Daniel, it's great pleasure to have you here today. And I leave the floor to you. Thank you again.
Okay, thank you so much. It's a real treat to talk to you all. And thank you for the kind introduction and an invitation. It's a real pleasure. My opening slide here, I hope you can see it behind me, is a picture of the United States, just to kind of orient you to where Williams College is. We're about 3 hours away from Boston to the west, and 3 hours north of New York in the mountains. And Williams College is a very special place. It's a top-ranked university, not university, but college. So we don't have a graduate program, but we have 2,000 outstanding undergraduates. And from here, they go on to, if they're interested in graduate study, on to some of the best universities. And I'll highlight some of the work that I've done with them in the course of this talk. I want to give you a little picture here of our kind of main quad. So it's a liberal arts college. So we have a very strong emphasis in the sciences, but also in the humanities and social sciences. It's a lovely place to be. We don't always get a rainbow, but sometimes. So this is a physics talk, and I have gone from transition sort of from statistical physics, using those ideas, using those verbs to apply them to biological problems. For your audience, I thought it would be wise to just sort of refresh your minds with some of the processes in biology, how we use mRNA to make proteins and so on. And so I'm going to tell you about some work that we've done about that has to do with increasing protein yield in bacteria and trying to understand those underlying processes, and then how that relates to the new mRNA vaccines like the Pfizer vaccine or the Mrna vaccine. My understanding is Pfizer is happening in Turkey. That's one of your options. Yeah. So we'll talk a little bit about that and what their design principles were and how vaccines work. So I'm going to start with the central dogma of molecular biology just to kind of orient ourselves. So DNA is, of course, the holder of genetic information and makes a double helical structure. When that double helix unwinds, it opens up one of the strands of DNA as a template for RNA, and then the messages in the cell are copies of one of the strands of DNA as messenger RNA. The messenger RNA then is translated into protein, and proteins are the workhorse of our cells, carry out most of the chemical reactions, and provide the structure. So this was some fine work by another Bachelor of Science in Physics star, Francis Crick, one of the co-discoverers of the DNA double helical structure. This is what, if you take an electron micrograph, in this electron micrograph going across horizontally is the messenger RNA. And this is the process of translating it into proteins. So there's a large complex called the ribosome, which is working its way along. And in this case, it's going from the right to the left. And you can see that as it's reading the message, it is translating it, and proteins are coming out the other side. In this case, you can see that the proteins just keep getting longer and longer as you move to the left-hand side of the frame. And that's how you can tell that the flow is off in that direction. So the ribosomes are working their way along the message in this picture. Here we're going to zoom in a little more closely. This is a kind of fun, wacky diagram of the message as it's being translated. So there are a couple of key components here. There's the messenger RNA. It is being translated 3 bases at a time by tRNAs, transfer RNAs, and the tRNA does base pairing with the message. But on its other side, it has an amino acid, and amino acids are the monomers, are the building blocks of the protein backbone. So here you can see the protein kind of emerging from the other side. So that's just a little bit about molecular biology. The way that the tRNAs decode the message is that they have 3 base pairs. So they're making 1, 2, 3 base pairs with the message, and then the next 3 base pairs will be read by the next tRNA, as you can see in this image. So those the rules are for the DNA and RNA that there are 4 different base types, A, C, G, and T. And if you read 3 of those at a time, then you have 4 times 4 times 4 possibility. Those are called codons. There are 64 different codons, but there are only 20 amino acids. So there's a degeneracy in the genetic code, and that means that alanine can be encoded in the messenger RNA with these 4 different possibilities. And one of the things that you can see here with the height is the relative probability of using each of the codons. And you can see that the codon usage is not uniform. Different codons are used more than other codons within the message. And sometimes those rules vary as you go from organism to organism. So in this case, you can see, for instance, that the GCG at the very top here, the GCG codon is used infrequently in humans, but more often in E. coli. So interestingly, the code itself is universal, but the relative usage is not universal. Another one maybe to highlight down here is the AGG, the black down at the bottom of the arginine, over to the right and middle of the diagram. I'm not sure you can see my mouse, is quite a lot smaller in E. coli than it is in human. Okay, so that's the relative codon usage. Amazingly, with the technology that we have now, we can get structures with atomic resolution of these giant complexes of the ribosome and the tRNAs as they go through their different sequential steps. So here on the left, the ribosome has the 3 tRNAs in there. So the tRNAs are recognizing the messenger RNA template. And then in the middle, you can see that the ribosome changes its conformation, and that change of conformation brings the amino acid in the tRNA, in the 2 tRNAs on the left-hand side in contact with each other, and that's where the chemical bond is shifted from the tRNA onto the growing peptide chain, the protein chain, and then it shifts a bit further over, making space for the next tRNA to enter. So there's this elongation cycle, and we've had a look at that. The protein data bank is a wonderful resource for some of these images, and gives you a sense of what's happening really on that molecular scale. So one of the things that I've been focused on is trying to predict RNA secondary structure. So I worked with quite a number of students on that project, and I'm going to just kind of give you a sense of what this looks like. So we've sort of seen the tRNA as those yellow blobs in the previous slide. This is what it looks like if you just highlight one of the molecules. You can see the backbone configuration here. And there are places here maybe where you can see planes of bases coming in and interacting with each other to form the base pairs. So this is the base paired structure. If you take a kind of simplified what's called a secondary structure, you have you can see on the right-hand side there a list of all of the bases. It starts up at the top with the GC, GG, AUU, and then finishes on the other side. And one of the things that this diagram helps identify are just what the base pairing rules are. They're the same as in DNA, more or less. That G's and C's pair with each other. So up at the top, the first base, that first G will pair with a C on the other strand. The C will pair with a G. Later we see a GU, which is a little bit less frequent. Also AUs and UA pairs, just in that top, the top part of the diagram. So those are the base pairing rules. And the RNA folding problem is to try to figure out how you can go from the sequence to figure out what is the structure, which bases will pair with which other bases. And my lab has worked on developing different algorithms based on the thermodynamics of RNA. But this is what tRNA looks like as a secondary structure, and then in 3-dimensional space when you can see where all the atoms are. One other thing that I'd just like to highlight about the tRNAs as we look at this structure is that this is just kind of a rule of thumb, but about half of the bases are paired and about half of the bases are unpaired and making up these kind of loop regions in between the pairs, okay? And one of the things that you may notice in the left loop and down at the bottom is that there are, and also at the right, particularly in those loop regions, you'll see a lot of modified bases, which have different letters. So there are Ds on the left, there are these psi and Ys and so on the bottom and on the right. So there are a lot of modified bases in the tRNA, and that turns out to be something that we'll come back to later in the talk as well. The messenger RNA also has secondary structure. In a lot of the diagrams that you see, it just is a long unpaired molecule, but in reality, RNA likes to form pairs with itself. And here's a structure that was determined for the SARS-CoV-2 genome. And you can see many of those same kind of hairpin structures or branch structures that are showing up here. And there are some ideas that these structures in the mRNA may be important for gene regulation as well. Here's a zoom in on one of those regions again. Okay, so same kind of idea, same base pairing rules: G's pair with C's, A's with U's, and you get these kind of interesting looking structures. So here are some of the students that have worked with me on these kinds of projects. We've done some work with RNA visualization to try to understand which bases are paired with which other bases and how we can define the local structures ensembles of structures that are sort of thermally fluctuating one to the next. So we've done some work on algorithm development using partition function kind of ideas where we include all of the states in our ensembles, and a little bit of math of counting the multiplicity, how many microstates we have. We've also done some work on structures. I'll talk less about this today with RNA pseudoknots. That's a particular class of structures, RNA structures that are important for enzymatic activity. Almost all of the RNA enzymes have RNA pseudoknots within them. We've done some work on polymer physics to try to improve the free energy models for the loop regions, and quite a lot having to do with the kinetics of folding as well. And then these are students that have been involved with me working on that translation process of trying to analyze experimental data about protein production and how that relates to the mRNA sequence features. So these are some of the students that have worked with me on that. Others have worked on splicing and photochemistry. So one of the real pleasures of being at Williams College is working with it's fun for me to see their faces again here. So we have a senior thesis project, and we really engage with our students to try to produce real research, and we find that this is part of what gives them a real leg up when it comes time to apply to graduate schools. Is they've had that strong research experience. So I think that's one of the special things at Williams that maybe people are unfamiliar with. All right, I'm going to transition a little bit to talking about how some of this research work relates to the mRNA vaccines. And as a little aside, I want to talk about what were the fastest vaccines that had been developed before the new round of vaccines for the SARS virus. The previous winner, so at the beginning of the pandemic, people were like, well, the fastest that we've been able to develop a vaccine is 4 years, and that was this Mumps vaccine. So I thought I'd just very briefly tell you the story of that. And it starts with the older girl in this picture, Geraldine Hilleman, who was the daughter of a guy who worked at a drug company producing vaccines. So one night she wakes up and she's got a fever and her throat is sore, and he recognizes that she has the mumps. So he comforts her, and then he goes into the lab and he gets his collection his collection stuff, some nutrient broth and swabs and so on. Comes back home, wakes her up, swabs out her mouth to get a sample of the vaccine, drives that back into the lab, and then takes care of her as she heals, and then goes on to spend this next 4 years trying to develop a vaccine. And in the center of this picture is his daughter being inoculated with the Mumps vaccine derived from her older sister's strain of the mumps. So mumps was a vaccine which was prevalent in the 1960s and prior to that, and there were about 1 million infections in the United States per year. For many people, it was a fever and swelling of the glands, but some people would have have complications, and in some cases it led to deafness and sterility. And so it's a fantastic thing that this vaccine was produced. So just to give you a little sense of this, its genome is 16,000 bases long, and it encodes 6 proteins. The proteins here take different structural forms, but some of them, the blue one and the pink one, coat the outside of the droplet and help it gain entrance into the cells. And others are responsible for helping the virus make copies of itself. And you know, that's more or less what a virus looks like. Sometimes they also have extra genes that help them avoid the immune response in some way. So here's more or less the plan. He's got Gerald Lynn's raw, you know, hot mumps virus, and the strategy that took 4 years was to take that mumps virus that's very infectious to humans and inoculate chicken eggs with it. So in those cells, the mumps virus turned out to be able to grow, but it grows not well. It is not well tuned to the environment of the chicken, to those cells. And so, what ends up happening is mutations start to accumulate, and the virus tries to, you know, the more productive versions of the virus that will grow the best in the chicken eggs are the ones that are more and more tuned. They're chickenified. They become more and more chicken-like and less human-like. And so the advantage of this as a vaccine is that then, but after 4 years of slowly accumulating mutations and making it less and less infectious to humans, but more and more infectious to these chicken eggs, then that version of the virus it turns out to still be able to give a good immune response, but it grows slowly and so can't cause serious disease. And so that's more or less the idea. The idea here is if you have the wild type, the fast growing, fast replicating one, it will grow very rapidly and produce a lot of copies before the immune system. So you can see that rise in the red curve there. It will produce a lot of copies before the immune system eventually starts to fight it off and damp it down. If you have an attenuated virus, it grows, but it's like, you know, the temperature is wrong, or the species is wrong. I'm just not growing as well as I used to grow. And so it will grow, and then the immune system recognizes it and beats it down again without causing anything near as high severity in the illness. So that's more or less the idea behind the old school vaccines. And Hilleman actually produced 8 of the 14 vaccines that are given in the United States to children, including the mumps. So the idea, again, is we're just going to give something to the immune system. The immune system doesn't know that it's not the full-blown real virus. It's going to build up its defenses. And once those defenses are there, it will be the immune system will be able to recognize those viral elements and fight it back. So even at a very low level, it responds very quickly. And this is the magic of vaccines. One of the things that's very cool is that you can go to the Gen Bank. So one of the courses that I'm teaching this fall semester is a computational biology class. And these are some of the kinds of things that we learn to do. So we can go to the Gen Bank, a resource that the NIH, the National Institute of Health, provides, and you can download the wild type mumps vaccine, the Gerald Lynn strain, so we can see what the sequence was of her strain of the mumps, and then the wild type mumps. And what you can see is there are many positions, most of the positions, 94% of the positions are unchanged, but some of the positions will have accumulated a mutation. 6.4% of the nucleotides have been changed. So that's the difference that it took 4 years to accumulate. If you then go and look at the protein sequence, so that protein sequence is what you get after you translate the 3 bases at a time, you see that there are even fewer changes. And that's because of the degeneracy in the genetic code. So a lot of times that final position, the 3rd position in the genetic code, will code for the same protein, but those are sometimes the things that produce differences within the organisms. So in this case, 1.2% of the amino acids were mutated. And this is close enough that the immune system will still recognize the Gerolin strain of mumps, the vaccine strain, as the real thing, and allow the immune system to fight it off. And what you can see here in this diagram, these big circles were sort of on the scale of 1 million infections per year. And after the vaccine, the orange circles tell you when the vaccines were introduced. And you can see that the prevalence of disease has dropped dramatically because of these vaccines. And so that means, many fewer kids have have a few days off from school with a fever, but it also means that smaller percentage that has the severe reactions, that those go down dramatically as well. I'm going to talk today about the SARS-2 virus and vaccine. And perhaps you've seen a version of this. On the horizontal axis, this is the infectivity. And you can see down at the bottom there is chickenpox and the mumps. Those were very contagious diseases, but diseases that were not very deadly. On the other hand, SARS here I've highlighted in the blue region, SARS-2. And what you can see there is that it's fairly infectious and can be quite deadly, particularly for older populations. So this was using data from the CDC. So older populations, the deadliness of the disease can be quite high. For younger folks, not so bad. Okay, so there's a big range there, and it may be that the single dots that they show for some of these other things are kind of aggregates as well. But we definitely know that the SARS-2 vaccine or SARS-2 virus has different effects on older and younger populations. Okay, so I want to just sort of zoom in on what the SARS genome looks like. Again, this is a thing that where you can get a look at what the proteins look like. And if we go along the top of the diagram here, this is just a list of all of the genes. There are some very big genes that are later broken up into smaller proteins. So there's what's called a protease that takes this very big ORF1 orange clump there and turns it into 10 or 16 different smaller proteins by making SNPs at just the right place. There's the spike protein, which is the first kind of big purple one there. There it is, and then some other ones that are part of the membrane and the envelope. So the spike protein is the part of the protein, is the part of the genome that as far as I know, all of the vaccines have focused on. So the spike protein is one of those surface proteins of the virus and helps it gain entry into the cell. And so when they're producing vaccines, they are not encoding any of the rest of the genome. It's only that spike region. So it's just that few thousand base long spike protein. You can see it's quite big, but that is the part of the protein that is part of the vaccine. So it can't replicate itself. It is only it's only the clothes of the vaccine, only the clothes of the virus. And so the vaccines present those clothes so that your immune system can recognize when somebody's wearing those clothes and fight off the real vaccine or the real virus when it comes. This is what it looks like, kind of zooming in just on the spike protein. And one of the things that's kind of interesting here is you can see that it actually adopts two different forms. So there's sort of a hinge in the protein that allows it to swing out, or it can take this kind of more triangular shape. Okay, here it is as it's gaining entry into the cell. It interacts with a surface protein of the cells called ACE2, which is expressed on our lungs, which of course is its kind of main target as a airborne disease. But you may have heard of some of the concerns about things with the heart and kidney and intestines, and it sort of makes sense that that happens because those are other places that molecule is expressed. So here it is as it's recognizing the surface protein. Also in the protein data bank, you can find versions of the spike protein as it's being attacked by antibodies. So there are versions, so you can really see where it is that the antibodies are interacting with the spike protein, and this is part of the mechanism of neutralizing the spike protein or neutralizing the virus, is to recognize those cells through these antibodies. So I'm going to now transition to talking about how you design a messenger RNA virus. And here we go. So here are the steps. So step one is you can, and once again, like the old Mumps vaccine took 4 years. It turns out that the mRNA vaccines only took a few months to design, and a few months more to test, and that's been a real saving grace. So the first step in this design is to just download the sequence, and amazingly, as soon as the virus was identified, its sequence was done. Sequencing technologies make that relatively easy, and it was posted to publicly available data sets. So the original virus was right there. As you know, the viruses tend to mutate, and we've been accumulating mutations over the course of time, and those now are the alpha and the beta and the gamma and the delta and so on, as the virus continues to evolve. So you can look at where the mutations occur and how they go on. As time goes along, viruses will accumulate mutations, and the mutations that help it become more infectious are ones that are going to win out. And so as time goes along, we tend to get more more infectious viruses. And here you can kind of get a heat map of where the mutations tend to occur. Okay, so we started with the original COVID-19 vaccine, and one of the things as I was preparing some of these materials, I found leaked to GitHub the sequences of the vaccines themselves. So of course it's easy enough to sequence them, sequence things if you can get a sample, and apparently somebody grabbed one out of the trash and ran it through the sequencer. So anyhow, it's out in the world. So I thought that was cool, and I just had a look at what was happening in the vaccine sequences versus the virus sequence. So we can compare the Pfizer-BioNTech sequence. Moderna is another company producing mRNA vaccines, and compare that against the COVID vaccine or the COVID virus. And what you can see is that actually all of it is only the spike protein that's in there, and the only place that there are any mutations in the protein sequence is these two bases that I've highlighted right in the middle of the slide. So I hope you can see that there's in the virus itself, it has a K and a V at this position, but in both the Moderna and the Pfizer BioNTech, there are these two prolines there. And I didn't know at the beginning what was up with that. So I did some digging around, and it turns out that that both of those companies follow the literature better than me. And they learned from this study in 2017. It was a study using the other coronaviruses, the MERS virus and the SARS-1. They studied the structures, and down in the bottom in panel D of this figure, you can see that the wild type sequences for MERS and SARS adopt those two different structural forms for the spike protein. So you can see the sort of triangular ones in blue, and the sort of more spiky looking things, pointy things in red. And what Jason McClellan's group realized was that by making those two proline substitution, they could take what was that hinge and lock the hinge into the conformation that the immune system has an easier time recognizing the protein in. So they stabilize the protein by putting in those two prolines, and that is good for training our immune response. So it's not it doesn't have to waste its time on a form that's not as prevalent. Second key feature that's part of the mRNA vaccine designs was some work that was being done in the early, well, in the 2000s and the 2010s. Carolyn Carrico was a leader in this effort. And so she was trying to understand what happens when you do gene therapy. So when you supply messenger RNAs to cells, a lot of their effort was in trying to design cancer therapeutics. And one of the things that they discovered is that they could design the messages and they could inject them, but they often led to an immune response, and would be quickly degraded. In a way, it makes sense that this happens because this foreign mRNA is very similar to what you would get in a viral attack, where we have foreign mRNA from the virus coming in. But one of the things that she discovered was that the immune response, so when they injected tRNAs into the cells, foreign tRNAs into the cells, that did not lead to an immune response. And she had the idea that maybe it had to do with these modified bases in the tRNAs, and that protected it somehow from the host immune response. So what they did was they just recoded the uridines, the U bases of the RNA, to pseudouridines, and they're quite similar. They form the same base pairs with the adenines. But two interesting things happened. It increased the total amount of translation, and it increased the mRNA lifetime, and reduced the immune response. So this was a kind of a magic thing. So in the mRNA vaccines that are out there, they're all using these pseudouridines in place of the uridines. And it turns out the other three bases don't lead to the immune response, to triggering the immune response. So just by making this substitution, they get longer lasting messages and more productive messages, more protein per message. Other things have to do with optimizing the sequence. Here I want to take a brief detour to talk about how our work in this area is similar, and some of the rules that we learned. This is work that I did in collaboration with John Hunt, who's a structural biochemist at Columbia University. Greg Boel was in his lab at Columbia, and now is an independent investigator in Paris. And so we were looking at what are the mRNA sequence features that control protein expression, And we got from work that John and his collaborators were doing as part of the Protein Structure Initiative. This was one of the follow-ups to the Human Genome Project. They were just trying to express proteins that people were interested in and trying to get more and more motifs, because one of the things that came out of the Human Genome Project was that we know that we have a lot of genes, but the function was not known. So just trying to generate more of these protein motifs was kind of a helpful thing. So they had done about 20,000 experiments, and about 6,348 of them were unique and became part of a data set that I mined to try to tease out what were the mRNA sequence features that were important. So we looked at a lot of different things and found many things had some statistical significance. I want to highlight just a few today. One of them has to do with the folding of the mRNA, and particularly right at the beginning of the message. And the reason that this is important is that, as we've said, that all RNA likes to have about half of its base pairs, half of its bases form pairs. But for the ribosome, as you've seen in those images, the ribosome can only bind onto the messenger RNA when the message is in its single-stranded form. So if the message can be unfolded, then that allows the ribosome to come in and interact with it and begin to begin to translate the protein. The ribosome itself has what's known as a helicase activity, which pulls apart the secondary structure of the mRNA as it's working its way along. So once it's engaged in producing proteins, it's burning energy and it can kind of clear the way in front of it, but it needs to initiate. So here in the blue squares in figure H there on the left, you can see that as the free energy increases, meaning less and less stable folding, that was highly correlated with increasing the protein expression. We also looked at the folding energy later in the gene, and there an average amount of folding seemed to be the best strategy. We looked at the base composition, and having base compositions that look as similar to E. coli's base composition was what turned out to be the optimal. So one of the things that we've done in the time since the publication of this paper in Nature is, we patented this process, and we've been looking at redesigning genes to try to maximize their expression in E. coli. Other features that were important were the codon usage, and some of the codon usage actually turned out to follow kind of its own rules. There are some codons that are more highly correlated with high expression than others, and so we could redesign genes to make use of the most productive codons. A last feature I want to talk about that relates to some work that I did with a couple of students before I got involved with this project. We were looking at the average folding energy. So this is a kind of an example of a mean field theory type exploration. So we were trying to understand what happens as mRNAs unpair. So we did some experiments, and we found that the different bases had quite different folding energies. And in fact, could be 2 kT different between the average free energy of a G base and the average free energy of an A base. So when I ended up looking at the protein production rate here, so here we're keeping track of the number of highly expressed proteins divided by the number of expressions with no detectable protein expression. And then we can keep track of what happens if we have an A at one of these different positions, or a C or a G. So one of the things that you can see here is up at the top of that left figure, those red dots are all when I have A's. And so having A's at the beginning of the sequence is correlated with high protein production, and having G's at the beginning of the sequence is correlated with low protein production. But you can see after about position 18, those rules squeeze together to be sort of more general. There's just less of a difference later in the gene. But this we interpreted this on the basis of the average folding energy as saying this is really the likelihood of having a structure that's going to prevent the ribosome from binding there is really strongly base dependent. So that's part of our design rules too. And again, quite a number of students have been involved in working on these RNA thermodynamics kinds of projects. And here are a few of their pictures. So with our metrics, we found that those model optimizations really work, and we could take genes where the wild type expression was undetectable, and then through a variety of different optimization strategies, turn it into a protein that was expressed as one of the highest expressed proteins in the cell. And amazingly, in these experiments, sometimes you can go from a situation where the, as you see here, where the genes, were undetectable, and they really can take over what happens in the cell. So it can be that about half of the protein production is your gene of interest. And of course, E. coli is still making a few of its own genes, but this is a kind of neat thing in biotechnology that you can take over E. coli cells and make them produce other proteins, and use that for crystallization or enzyme production or various other kinds of things. At a kind of around the same time, Mrna was doing some of its own studies with human sequences, and so they were trying to figure out how to design redesign different proteins, what controlled their stability. So once again, kind of the key ideas are to increase the relative synonymous codon usage, try to make the genes look more like human genes. And then they also looked at what happened with GC content. And Mrna had an idea that other start sites, like the messages can sometimes be translated in a variety of different ways. And so we'll see that one of their strategies is to eliminate the alternate start sites. So this was a paper that was published early in 2019. So before the pandemic had begun, they were already kind of figuring out what their rules were. And so that was part of what made fast action possible, was that then all you have to do is plug in the new sequence and go on to the next step in your testing. So if we compare the virus, we see that the virus is not highly correlated with the codon usage in humans. And you can see that the correlation increases dramatically in the Mrna and the Pfizer-BioNTech versions of the vaccine. Other things that we looked at were just for fun, since I was interested in RNA folding energy, was what happens with the folding, and in both of these cases, they maximized. They seem to be trying to increase the overall folding stability of the message. And Mrna had that as a particular design principle, and you can see that in fact its free energy, its folding stability is the most stable of these three options. Here on the left, we're using a visualization approach that a student and I developed called RNAbows, where we can highlight with arcs. These arcs here connect which bases are paired and which bases are unpaired. And I like this difference RNAbow approach to help highlight the parts of the sequence parts of the structure that are the most different. So you can see these base pairing arcs are sometimes the same when they're colored in black and sometimes different. Okay, so in the end, what you can do is you can translate the message and look at it in the different frames. So if you just look at the COVID genome, you see here is the spike. It's possible to translate that 3 bases in the 2 other reading frames. And the pink part here is highlighting the long open reading frame that actually encodes this, the spike, but you can see that there are some places where it might, in frame 2 in particular, where there are some start codons, starts with an M, and will go for a little ways before encountering a stop codon. If you compare with the Pfizer-BioNTech, again, the actual spike protein is almost identical, but you can see that the frame 2 and frame 3 translations are slightly different, and that's reflective of what's happening with the base changes as well. If you compare with the Moderna, one of the things that you see here is that they really have eliminated all of the out of frame start codons. So the frame 1 is the only open reading frame that's possible here in their design. Once you've designed your spike sequence, you encode it in a little bit of some bookends that help it express in the cells. So it needs a untranslated region at the beginning and the end, and also this signal peptide that directs it to the part of the cell that it needs to go. So these are sequences that are out there. And then in the next step, then you have your mRNA, and you've grown this up with the pseudouridines, and you've put in your you've got your sequence ready to go. Then you need to figure out a way to get the body to take it up. So another large effort that happened in the 1990s, 2000s, 2010s is working on lipid nanoparticles, and these are things that bind to the mRNA. Lipids are sort of like soap type molecules, so they'll sort of present themselves as kind of fat globules to the cells, which helps them be taken up. Here they're encapsulated in polyethylene glycol, and then they've got the mRNA right in the center. So a lot of work was done to figure out how to design the lipid nanoparticles to deal with these rather large messenger RNAs. And they're just like 2 or 3 of the messenger RNAs per globule. But because they present themselves as looking like fats, fats, our cells are happy to take up fats for nutrients, so they get engulfed and brought into the center, and it's only there that their cargo gets released, and then our ribosomes begin to translate that message, and it goes from there. So once you've got these technologies in place, and all of these technologies, interestingly, were kind of coming into fruition just a few years ago. Then it's time to figure out how to deliver the vaccine. So now we get the vaccines, and it's getting the shots in arms, and once the shots are in the arms, biology takes its course, and we begin to translate those messages. So the testing then could happen, and it happened relatively quickly because there were so many infections going on. But the data is really quite amazing. So here we have on the left the Moderna data, and on the right is the Pfizer data. And what you can see is the placebo, so a group that didn't get the messenger RNA vaccine, but just a saltwater substitute, continue to contract the disease. People who got the mRNA vaccine after about 10 days, you can sort of see that the curves, particularly on the right there, you can see that the curves are about the same for the first 10 days, and then it just goes flat. So the vaccinated population, their incidence of contracting the disease or coming down with a serious version that required hospitalization, it drops by an enormous factor of 30 or so for 2 vaccines. So in the end, part of the song in my heart is that we get to celebrate these results. And I just love this cartoon. It made me tear up a little bit, to be honest. You know, it's like when the data is this good, that you don't really need to do any kind of statistics to tell the difference. You know that you've got something magical happening, and thank goodness we're in that situation now. So just to kind of wrap up, in 2020, it took 2 months for Moderna to go through the process of figuring out what the mRNA sequence was going to be, and formulate it for the delivery, and do some initial testing to try to figure out what the dosing was, and get it out for phase 1 tests. And if COVID had struck a few years earlier than that, it might have taken years. But because basic research kept chipping away at these projects in the meantime, it was possible to very quickly develop these vaccines. And as we've seen, they're beginning to transform the world, open up the world again for the way that we would all love it to be. So those are part of the joys. So I think we have to just keep investing in basic science and keep training the next generation. And I'm glad to be part of that enterprise, and look forward to having some of your excellent students join us here at Williams College. I'm happy to take any questions at this point about any of these topics. So thanks so much for attending today.
Thank you so much for this very interesting presentation. And also thanks to the whole scientific community for this miracle that has happened just in the graph that we've seen. So Nihat Hocam, Sondan Hocam, would you like to start?
Yes, I have. Daniel, thank you for the presentation. And I have a question about, yes, Pfizer and Moderna, the mRNA vaccines, they are working. I don't know if there are statistical calculations or others telling us how long they are protective. This is a discussion on when you saw this, the graph, yes, with the vaccination.
Right.
Yes, it is an excellent result, but how long that's gonna
right.
I don't know. Do you have any idea? I don't know. Yes, we follow the newspapers, but I want to hear from you, Danny.
Yeah, there's been a lot of discussion with that. I think the original idea was that it might provide relatively long immunity. Yeah. But now people are beginning to get booster shots. Yeah. Apparently there's some idea that it begins to fade a little bit. And I think we won't know for sure. We won't know for sure for a while what the facts are with that. Yeah, but it is an interesting question. Yeah. I think at least here in our country, the process once vaccines were available of getting them to the frontline workers and older people, prioritizing from older to younger was a correct decision.
And Daniel, is it, I don't know, I just, that may be quite naive question, but by looking at the structure of the mRNA vaccines, and can we have the predictive power on that issue? I mean, just structure, investigated them, and scientists, can they tell this could be protective for that long period?
Oh, I see. Yeah. So, okay. Yeah. So I think I think just looking at the molecule is maybe not going to be quite enough for that, because a lot of it has to do with other processes in the body that have to do with how long do the so it's not about the molecule anymore. But interestingly,
molecules,
yeah, it's more about the antibodies and how long the antibodies persist. Okay. So I was having a discussion with one of my former students who's now a computational biologist at the in Boston, and they were trying to decide, should they get, they'd had Pfizer, should they get Moderna for their second shot or the other way around, and the protein right now that is being produced, the spike protein is the same for all of them.
Yeah.
So at that level, I don't know that there's a difference, but yeah. So there's other interesting modeling kind of things maybe that could be done to understand what happens. And I think it is. I've heard a presentation that there aren't that many of the different antibody molecules that are floating around in our bodies. Mm-hmm. It might be on the order of 1 million, particularly right after the infection. But like whether they stay or go, like we have 1 million for this infection, we have for this virus, 1 million for that virus, 1 million for that virus. Is we're exposed to so many different viruses, but it does seem to produce some kind of protective effect. Sometimes even a long time after. As a father of young children, I know they came home from school with sicknesses that didn't infect me again, because I probably had them when I was in elementary school. So
yeah, thank you, Daniel.
Yeah, thank you. Yeah. Thanks for that question,
Daniel. Thank you very much for a heartwarming talk, for taking care of problems, for being prepared for them, and the message in favor of basic science, and for a great rendition of this. Thank you very much.
Thank you.
Our participants here and our viewers on the YouTube live broadcast, if you have any questions, you can type it into the chat box and I will read it for you. Or anyone would like to pose a question or a comment? We have some claps from our professors. Thank you, Underhocam.
So Daniel, what is the undergraduate application deadline for Williams College?
Okay, I think I'm not 100% sure, but I think around the new year. There are some early decision that deadline may have passed by now, but
that has passed, I know.
Yeah, but I would guess around New Year's.
And how many students do you take each year?
Yeah, so the overall population is maybe 2,200, so 550 per year.
And what this is a third degree. What is roughly how of that 550? Roughly how many are international students?
Yeah, we have quite enough. I guess I didn't say this, but we have about 10% maybe that are international students, and so there's been an effort to grow that population. And one thing that's true about Williams and many of the other top liberal arts colleges is there's very strong financial aid. So students have their bills taken care of for them, to allow all those top students to come here. So that's been a real blessing.
So if there are questions from our students in Turkey, they can send them to me by email or phone, and I can pass them on to you, right?
Okay, sure.
I have a question.
Yeah, wonderful.
Yes, Daniel, thanks a lot for the nice talk. So I wonder if you compare this Chinese vaccine, the Sinovac, with this BioNTech and Moderna, what would you say about that?
Yeah, so the versions that are the closest to my research have been the Mrna vaccine. So I end up going to conferences with some of the people that that are key players from Moderna in particular, as a Massachusetts company. I think the Sinovac is a vaccine where they have another virus, and then they've sort of used molecular tools to splice in the spike protein, kind of in the background of some other virus, and so that mechanism of action is maybe a little bit different. It relies on, but I'm not 100% sure. There are many different kinds of many different vaccine approaches that are out there, but some of them use adenoviruses and just replace a part of a relatively innocuous virus with this spike protein. And again, the idea is just, by presenting that spike protein, it trains the immune system to recognize the outside of the virus before it can really infect and cause trouble.
Okay, thank you. Yeah,
thank you.
Mm-hmm.
Thank you. Any other questions or comments from our participants? You can type it on the chat box if you'd like. Nuray Hanım, I am unmuting you right now. There you go.
Thank you very much. Thank you very much for this nice presentation. I have a question. There are certain sequences, as you may know, these internal ribosomal entry sites, T2A structures. They allow ribosome skipping, so we can produce multiple proteins from one mRNA strand, let's say. Can we use these sequences in this type of mRNA vaccines so that we can produce many proteins of viruses? Or will it be create too long mRNA sequence which disrupt some events?
Okay, yeah, no, that's an interesting question. So I think one of the, I'm going to answer it in two ways. One is that I think if you have a good idea about what the internal ribosome entry sites are, those could be even more important than just having a start codon. So as part of the design pipeline, you might want to make sure that you are not creating an internal ribosome entry site that would produce some other partial products. So that could be one answer. Another thing that I'm thinking of is one of the challenges with the spike protein is it's just relatively big, and so I think the idea of trying to figure out how to encapsulate a minimal set of proteins that will produce a robust immune response has kind of been the strategy. So rather than producing the spike protein and the membrane protein, and some of the others on the surface. It's just been that one protein. But these companies are, of course, going out and doing some work trying to figure out how to cure other viruses as well. So, like you could imagine trying to do this for the flu, and in that case, there's the H1N1. So you want to present maybe the H protein and the N protein, and whatever the different variants are there. So that could be a place where that would be important. And one thing that I heard, this was preliminary results, but one thing I heard was that there's some promise that the mRNA vaccines might permit kind of broader immunity against the flu proteins, because they allow you to encapsulate the whole surface protein, and so there seems to be some recognition to the sort of stalk part of the protein, and so they were getting cross immunity, and I forget the details, but like where the H1 and the H3 and the would all have some antibodies that were protective more broadly. So maybe that seasonal flu shot could become less, somewhat less frequent.
Thank you so much for the question and the answer.
Yeah, wonderful question.
Do we have other questions? There are some thanks from YouTube as well.
Okay, well, thank you.
Great talk. Barış Tamer Tonguç, thank you so much. So if we don't have any further questions, I would like to pass the mic to Nihat Hoca and Sondan Hoca for their last words.
Well, thank you very much, Daniel. Excellent. Great to have you with us. Sondan Hoca,
thanks for having me.
Yeah, I leave the word to you. Last word.
Maybe before we leave, Mustafa Hoca would like to have the word, I think. He's just written to me.
Thank you, Yeşim. Sorry, I was trying to unmute myself because it wasn't allowed, I think. Anyways, thanks, Daniel, for the great talk. Sorry, I was a little late because of some work I have to catch up, but I was able to catch up 35 minutes of your talk. One thing I will ask is, it's maybe a little off topic because you usually show the mRNA vaccination prep and then how it's really built. But as a person who got 2 shots of BioNTech, I had the numbness problem. So like my face all the way up here, and then my arm, I had right after the shot, like 2-3 hours after, I had the numbness problem.
Wow.
So is that something normal? Because I look at a couple details from the World Organization and the NIH. I didn't really see a lot of details about this. Is this normal, or it's just my body reacting against the vaccination?
Yeah, I haven't heard of that as a side effect, and I would have some caution, I guess. So some people, I think it's relatively rare, but some people have a reaction to the polyethylene glycol that's part of the formulation. So there are some things to kind of, I might be careful about that with the future. One of the things, when we get our flu vaccines, we have to kind of check off that we're not allergic to eggs. And, that, now, we can understand why that happens because they develop those vaccines in chicken eggs. But there can be some of that kind of, cross immune response kind of things. If you have an allergy, maybe to the polyethylene glycol or to eggs, you know, those are things to pay attention to. So I'm sorry that you had that that you had that reaction.
Yeah, it was kind of weird. And these pack chains are used for the solubility purposes, right?
Say again. Sorry.
Is the pack chains like the PEGs, they are used for the solubility purposes or what?
They're used, yeah. They increase the solubility and prevent the globules from kind of coalescing into bigger globules.
Okay, thank you.
I think, yeah. So trying to figure out that formulation so that you get a relatively small amount. I mean, it's amazing that it's 30 micrograms or 100 micrograms dose of mRNA. And it's in there doing its thing for 6 hours or something like that. So that's quite different from getting the full virus and being sick for a week. It produces those proteins, and the immune system is like, oh, well, that's not good. Let's figure out how to fight those off. So it's a relatively small amount. Even less in the new, I don't know, here the kids from 5 to 11 are have just become eligible for the Pfizer vaccine. So there it's just 10 micrograms.
Thank you.
Okay, thank you.
Thank you so much, Sandan Hocam. Your microphone is off. Can we turn on your mic?
Okay, yeah. Thank you, Daniel, for the presentation and for being with us today.
Thank you again.
You have a healthy life for the coming months.
Best wishes to all of you. Yeah. Thanks again for coming.
Yeah.
I would like to thank everyone, especially Professor Aalberts, and all our participants and viewers today. We will continue with our traditional public colloquia in 2 weeks. Again, please keep following us on our social media accounts and our website. Thank you so much. See you next time.
Bye-bye.
Bye-bye.